In this video, we will discuss about enhanced CD19-CAR-NK92 cytotoxicity against CD19 positive target cells. So basically, they will compare the cytotoxicity of these CD19 positive CAR-NK2 and the wild type CAR-NK2 cells. So for this, they started with this wild type CAR-NK2 cells and then expressed CD19 CAR using lentivirus transductions, and they validate their lentivirus transductions using the flow cytometer results that is shown in the panel A. Here we can see that 98% of the cells are now CD19 CAR positive, and thereafter what they did is they took different target cells, and some of those target cells are mentioned here, or the target cells are mentioned here, where they also check the CD19 expressions in these target cells. Except Jurkat cells, CD19 expressions are found in all the other target cells, that is Dowdy cells, CARpus 422, primary B cells. But Jurkat cells does not has CD19 expressions, or they are CD19 negative cells. Thereafter, once validating the CD19 expressions on the target cells also. So in this case, the primary B cells are from the patients that from which they get these primary B cells. Otherwise, all three target cells are the cell lines. Thereafter, they did the coculture assay, where they co-cultured the wild type CAR-NK2 and the target cells, and in the other case, they co-cultured the CD19 positive, CD19 CAR positive NK92 and the target cells. And then after co-culturing them for 4 hours, they check the target cells death or target cells lysis and compare it between the wild type NK92 co-culture and the CD19 CAR-NK92 co-culture, and they are able to see that for target cells that has the CD19 positive expressions, the cell lysis is increased when they co-cultured with CD19 CAR-NK92 cells. That can be seen from Dowdy cells, that is increased from 26 to around 90%, and for this CARpus 422 cells, that is increased from 10% to 50%. Primary B cells has also increased from around 30% to 50%, but the Jurkat cells, the increase in the cell lysis is negligible, and the reason is that the Jurkat cells does not have CD19 expressions, so thereby there is no enhanced cytotoxicity effect against the Jurkat cells when they interact with the CD19 positive CAR-NK92 cells. And so in this case, the mechanisms by which why this enhanced CD19 CAR-NK2 perform better than the wild type NK92 is not being discussed here, but that will be covered in the subsequent video. And researchers in the first figure just presented the point that the cytotoxicity of the CD19 positive CAR-NK92 cells is more compared to the wild type NK92 cells, and we will discuss in the subsequent videos that whether this cytotoxicity effect is because of the lysate or lytic granules that are released from the NK92 cells once they are activated, lytic granules that is perforin or granzyme, or it is because of the receptors, FAS receptors or that cell receptors that are been activated on the target cells once the NK92 cells interact with these target cells, or it is the combination of both. So these are just so far we can say that the options that are available with the researchers at this point of time, and how they evaluate these options, and whether their their speculations of one or whether their speculations of these assumptions are true, or they are able to find out some new pathways or new mechanisms by which the CD19 CAR-NK92 positive cells triggered lysis or the cell death of the target cells. That we will discuss in our subsequent videos. I hope you understood. this simple experiment to validate the enhanced cytotoxicity of CD19 positive CAR-NK92 compared to the wild type. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.